All right, I think we're going to get started. I hope everybody enjoyed lunch, and we are closing in on the home stretch of this meeting. So as I said earlier this morning, this part of the program is really focused on the drivers for adoption, and the next talk and then the next panel discussion will really represent the funders and then followed by the journals. And certainly in the United States, and really the entire world, I think, there's no bigger funder of basic biomedical research than the National Institutes of Health, or NIH. And we are very fortunate today to have with us at the workshop Dr. James Anderson. As Deputy Director for Program Coordination, Planning, and Strategic Initiatives, Jim reports directly to Francis Collins, the Director of the NIH. The division that under Jim plans and implements several trans NIH initiatives supported by the Common Fund. Prior to joining NIH, Dr. Anderson was professor and chair of the Department of Cell and Molecular Physiology in the School of Medicine at the University of North Carolina. And before his appointment there, he was professor of medicine and cell biology and the chief of the section of digestive diseases at Yale Medical School. He himself has actually produced many antibodies for use in his own research programs, so he's a member of the club. And he's also purchased several, he tells me. Jim is in a unique position in his role at NIH to describe NIH's effort to enhance the research and the Rigor and Reproducibility initiative that many of you have heard about and maybe even experienced when you've recently written your own R01 applications, and the Common Fund Protein Capture Program, which I think Jim will talk about. And NIH's experience with validating products from this program. So, Jim, it's a pleasure to have you here. Thank you.
Thanks very much, Len. Thanks for being able to provide a perspective on if we could go to the first slide, on the issues of rigor and reproducibility in the research that's funded by NIH. It goes without saying that this is fundamentally important to how we think about the research. If it's not rigorous and reproducible, it's not valid and useful for the purpose of advancing science. And I think one perspective that we have to put on the table that's not come up yet is we owe it to our patients to make sure that the research that we do is rigorous and reproducible so that it leads to safe and effective preventive measures, diagnostics, and therapeutics. So there's really a lot on the table here. And thanks for being able to be involved. I realize that what I should do before I start, though, is give a perspective that would emerge eventually, and that is NIH is not a regulatory body. We cannot credential people to do or not do research. We cannot set the standards and hold people to them. In fact, if there are regulatory issues, we step out of NIH and the USDA, who carries guns, by the way, they can step in and deal with a violation of animal issues, or the FDA can step in and deal with human subjects protection issues. But NIH is a research organization, and we don't want to limit people because we want everyone to be innovative and think of things that we don't do, not think of things that you have to do. So that's the perspective that we take to it. Now, we also have workarounds, and part of it is if we could come up with valid standards, we can hold reviewers to using them. Okay, so that's why this is very important that something come out of this workshop. Okay, this has jumped ahead a bit, but so the issue of rigor and reproducibility is obviously a long-standing one in science, and that's what we aspire to. I would say 3 or 4 years ago, there began to be a series of reviews of the literature that pointed out the tendency of preclinical trials to fail when pharma tried to pick up on a target or validate or reproduce a large study from a lab that pointed to a new therapeutic direction. And this got in the press. It got in the scientific literature, Science, Nature. It rippled out into the near-term valid people we think of that pick up science, like the science writers at the Wall Street Journal and New York Times. And it even got out eventually into the lay press here. So it became pretty well acknowledged, and people were admitting we have, as someone said, we have a problem, Houston, that we need to deal with and address. It's also clear that it occurs across all the areas of science. It just surfaced and became very relevant in the preclinical research, particularly because the next step would be a human, and potentially hurting a human if this wasn't a valid approach. Okay. So what were some of these deficiencies that were noted in research design and the way research was implemented? It was often insufficient reporting of the details in the publication so that they couldn't be repeated. I can remember, I'm old enough, I remember when Cell came out, and I was shocked this was a journal that moved the methods to the end of the paper? What message was that sending? Anyway, others, p-hacking I want to talk about. Other things like what we're really putting an emphasis in my division is the Office of Women's Health that's promoting the importance of sex as a biological variable. We're in an age of precision medicine, and yet we have organisms, humans that differ in their chromosomal composition, and that's not being taken into account often in our research. So that's often another cause of variability or loss of reproducibility. So one major problem, obviously, and we haven't taught our students and postdocs sufficiently, is the appropriate use of statistics. And one odd example here is what's called p-hacking, where in a large study you reanalyze the data or aggregate different parts of it until you find something that's statistically significant to publish. And I think just in general, there's a very poor understanding of the use of statistics and power design by our students and by investigators. So here's an interesting review. It comes out of back from our National Institute of Neurologic and Diseases and Stroke. They became very interested because so many of their preclinical mouse models of transgenic stroke studies, stroke pathophysiology, Parkinson's, multiple sclerosis, could not be duplicated by pharma. And again, the implication is if you could hurt people if you advance these ideas that were not true or could be dangerous. So here's an example of an analysis of hundreds of papers with different mouse models, and then asking the question, did they randomize the subjects to different arms of a study? So they could have taken a bunch of littermates and done the control 1 week, and a week later come back from a different room and took the littermates to do the study. So they've introduced multiple variables into the experiment that they're not accounting for. Another one that's come up already is the lack of blinding in reading the data, reading slides. I mean, it's never done. And then the most egregious problem here is, as you would require in a human clinical study, is an analysis of the power that's required if you expect a certain effect size. And that's basically never done in preclinical trials. So many problems in study design that lead to a lack of reproducibility. But the one we're focused on here is the difficulties in validating and using antibodies. We've already seen this review. This is from last year. It points out the large number of companies involved that provide varying levels of validation or data to the user, and what a large market this is, which could be a driver for some of the problem. And then an overall, just putting together various studies quoted in this, some have speculated that about half of the antibodies produced are not valid. Now again, as what's come up, what does that mean? It can mean anything from a denatured protein to the investigator doesn't know how to use the product that was delivered, that they purchased. But anyway, it leads to invalid research. So putting this all together, it's about 2 years ago now, our director and principal deputy director, Larry Tabak, decided it was time for a commentary and a set of actions at NIH to address these problems. And I guarantee you we take this very serious. And it was the result of some of the recognitions that I pointed out already, and then the statement that we were going to take some systematic changes in how we address these within the policy authority that we have. And again, this is not regulatory policy, but what could we do? So the things that we're doing fall into really 5 major categories. One is just to raise community awareness, and I'll give you some examples. Enhance formal training of what it requires to do rigorous and reproducible research, and I'll give you examples. And in this one, which some of you may have encountered if you've submitted a grant application this year, is adopting more systematic review processes. And I'll explain what that means and what the implications are. And then sharing information and data. This is a major issue for us. The culture comes from the Genome Project, where they would make data available daily with a download, and it would set a new culture. We have dbGaP now. We're pushing this in many ways. Last week, a policy was put in place that if you do a human clinical study, you must publish the data or provide it to the public within investigators, if it's identified data, within 12 months, and if you don't, there are very steep fines. But then again, who levies the fines? It's the FDA, not the NIH. Okay, but this is a serious issue. We generate all this data now, huge amounts of data daily with omics and genomics, and we have to have access to it, and others have to reuse it so it's not wasted. And we also, this has come up, someone's even used our favorite term, which is perverse disincentives, or that we have created what you might call the biomedical industrial complex by our overfunding and the commercialization of how we do science with kits rather than thinking. And it's created the approach that you have to have a Cell Science or Nature or CNS journal paper to apply for a tenure track job. And even the postdocs at NIH that I talk to, they think they need a cell paper before they can leave and try and look for a job. That's just not right. Okay, I'm going to take an aside because there's an office in my division called Portfolio Analysis, which really has a bunch of Google-like people. And if you check, we published a paper last week in PLOS Biology promoting a new metric to replace impact factor. It's called relative citation ratio. And it's a measure of the article level influence of an article, not the journal you published it in. And it's based on looking at citation rates within co-citation networks. And we've gotten a lot of interest. Wellcome Trust is applying it to all of their grantees. Many of our institutes are picking it up as another way to say whether publications have influence in their fields. So RCR, PLOS Biology. So we're looking into ways to increase the stability of investigators and try and reduce some of these perverse incentives that we've set up for over-competition. Okay. Okay, first of all, just a few examples of raising community awareness. And this goes back a couple of years where we set up a series of workshops with pharma, with journal editors to try and see, we can't force them to do it, but we can bring people to the table, make them aware we have a problem and try and coerce them into action. And this particular workshop led to a commitment from 135 journals to enhance their review criteria. And you've probably seen in recent years, and I guess Cell 2 weeks ago, a set of specific criteria that they expect reviewers to address or applicants to anticipate will be reviewed. So I think that's helpful. Journal of Neuroscience, very specific about are the statistics appropriate for this figure. And that comes from the interest in our neuroscience institutes because of the lack of translation of neurologic studies to clinical application. Okay, enhancing formal training. Now this is also where you could help if you can see where recommendations could be made. Again, NINDS has been a leader for us and our intramural research program in designing training modules, and these are available online if you choose to use them. We're using them to pilot an intramural program. We have thousands of postdocs at NIH. and hundreds of graduate students shared with other programs. So we are developing these and using them. They're available. NIGMS has also been a leader here, and with other institutes has awarded grants to develop training modules in this area, and also issued some administrative supplements to existing awards if it's within the scope of the award to develop training activities. So this is in the pipeline. I can't show you what these look like, but there's an effort to do that. If you could think of ways that different societies or antibody focus groups could promote training modules, that would certainly be helpful, and if we could find a way to make them available for trainees, then they might be effective. We also, in the intramural program, have had quite a number of workshops that have addressed potential pitfalls in specific areas, like FRET. FRET is terrible if you don't know how to do it and interpret it. Super resolution is fraught with problems if you don't know what you're doing. And so are antibodies. And these are available here, and I will also show you an example of a workshop addressing antibodies. I'm not getting this to, oh, there we go. Here's an example of a workshop that's being led by Ken Yamada, who you might know. He's an intramural person who is one of the editors of Journal Cell Biology, and he's addressing all the problems from beginning to end that you can encounter in using antibodies. And I just pulled out one of his slides. Here's presumably a specific antibody by Menton, but the issue here is fixation and permeabilization leads to a non-specific signal versus probably the specific signal. But you can also, you can have all of these training modules are available online. How about the issue of stability? Again, we have set up an incredibly competitive environment that's probably not productive anymore. So NIH is piloting some new award mechanisms that try and reduce that. We don't have a central approach. Each institute is doing it in their own way, but the basic principle is to fund at a higher level for a longer period of time and not hold individuals responsible for specific aims. And so basically it's actually, I'm an intramural investigator and we don't write specific aims. We're reviewed after the fact. And so we can come in every day and say, oh, that's what I should be doing, not what I wrote in my grant. So there's a lot of flexibility and you can really pursue what matters that day. So that's the basis of these grants. It's not holding people to specific aims, but funding the lab. A really interesting one, general medicine intends to push way over from R01s to this mechanism in R35, which is longer time, more money, but it's a lab grant. It's not a very specific set of specific aim grants. So this will be part of the portfolio. R01s will always be the major mechanism, but there are experiments to see whether we can reduce this intense sense of competition. Okay, now the interesting part, and I hope you can help us here, and these are changes to the review process. And these occurred on January 25th of this year. The Office of Extramural Research issued new guidelines for how applicants write their grants and how they'll be reviewed. And they're based on the following steps. First of all, we're requiring that applicants put their own project in the scientific, state the validity of the context that they think they're putting their work into. In other words, the scientific premise of the project. For example, I have reviewed the literature and I consider the following points to be valid, and based on that, I'm taking this next step. Or you could even say, I've reviewed the literature and it's completely worthless. Nobody knows anything here. And so that's why I'm proposing this. But it's putting it within what's the scientific premise or context for your work. There's also a requirement for review that there's a more rigorous explanation of the experimental design, and this would be things like power calculation statistics, a consideration of sex as a biological variable, and then an unscored criteria. This is a separate document to be filled out, it's very brief, on authentication of key biologic and chemical resources. So I want to spend a little more time on that because that's where the antibodies are. And this has been in effect since January 25th. I've talked last week to the director of the Center for Scientific Review, and it's really too early to report to you how applicants have responded, how reviewers have responded. And then, as again, it's not a reviewed criteria, but I'll just go ahead here and show you. This is what you'll see if you go to the website, the description of what's now required. And among the new attachment to be included with the grant is how you will validate these reagents. And they include cell lines and antibodies and some other issues. So I want to step outside and say, where did we get some of our thoughts on what we needed to require? Again, we're not a regulatory body, so we looked at other agencies within Health and Human Services that are. And for example, the Food and Drug Administration, their CEDAR division, this is the group that has to approve biologics. And what is the basis for it? They have a heavy reliance on reliability and reproducibility for the intended use. Set of specific criteria here, but again, method are suitable and reliable for the intended analytic purpose. So we don't set it ahead of time. We work with the investigator, in our case with the applicants, to say, how are you going to use this? What are you going to use to know that it's valid to interpret the results of your experiment? And so that's really the design that's built into the background to the guidelines that we now use at NIH. And these are on the web, but I want to read them. The applicant should briefly describe the methods they plan to use to authenticate resources based on their scientific experience and judgment. This is that's a little vague. I'm not sure how reviewers are going to interpret that. It could be a matter of if you have had 30 years of R01s and trained 50 postdocs, the reviewers can hold you to a higher standard, but I think there is a standard and everybody needs to meet the standard. We're encouraging the scientific community to establish consensus standards. And also, I think this is key for considerations here. Even if you purchase something that's, quote, validated, we are asking our reviewers to review whether you have provided your own authentication plan. So I bought it. It had this gradation scale that we've talked about today. But in my own lab, maybe that influenced what I purchased, but I'm still responsible in my own laboratory to validate it for my purpose for this experiment that I've just explained to you in the specific aim. And then a very important issue is we're not asking for the authentication, we're asking for your plan to authenticate in the application. So this I pulled off of a training slide that's being used for the reviewers. And the key issue down here is authentication of antibodies. Reviewers will comment on brief plans proposed for identifying and ensuring the validity of those antibodies. And they're being asked to rate as acceptable or unacceptable. And this is not supposed to influence the scores. But if it's unacceptable and the score and the overall impact score is within a funding range and the institute wanted to fund it, because as you know, we don't fund in order. Sometimes there's skips to meet priorities. But if the institute intends to fund it, then the program officer has to contact the investigator and say, study section didn't think you had valid approach or plan to validate. Let's talk about that. What's your understanding of what you would do? What standards might you use? Are you able to do this? Can we work this out? If they work that out, then the grant would be fundable. So that's an interesting step. And I think that would be very, very helpful for our program staff to say, oh, did you know that the such and such a society has standards that are highly relevant to your experiments? Can we go through those together? Are you able to do this? Okay, then we can fund it. Or no, we don't know anything about that and we don't know how to do that. And there goes your grant. So there's several steps in here where we really could, and our reviewers and program staff could use some standards from the community. Okay, I wanted to finish just with this as an example of a project. And I bring it up because it's in the Common Fund, which is in my division. This is a program that was first funded in 2011, and it was begun with a very audacious goal of creating some type of capture reagent for all 20,000 reagents in the genome. The first few years of funding supported alternative capture reagents, exploring. Well, the question was, can we scale this up within the 10-year limit of a Common Fund program to do this whole project? So it had a pilot-y sense at the beginning, and it funded some novel approaches, like at that time aptamers. There were even some chemists who came in with funny polymer surface mod covering things that didn't go very far. So actually the program reverted to recombinants and monoclonals fairly quickly, and also because it seemed unrealistic to know that 20,000 could be done immediately. It also reverted to transcription factors and associated proteins. So there were centers set up to make the immunogens, to make the monoclonals and recombinants, and to make them available for the community through a website that had done some level of validation. So here is what's now resulted in a portal that you can go to learn about these reagents and order them. There's over 17,000 reagents to a large number of transcription factors and their proteins. You can pick an antigen, not an antigen, I'll say a gene, and then the antigen within that gene, and go through the different steps in the production and validation and learn how they did it. And it was based on a two-step validation. And I would call this I put in this word screening validation because, again, it's the user who has to do the final job. But the first step is a primary screen for specificity, and this was things like array. I think there was some plasmon residence used. And if it had some specificity, it was then applied in a second step for potential application, and that would be things like immunoblots. So you can pull the data out of there, and as I look at it, I'm not in the transcription field area, so I just looked at it if these were cell biology relevant antigens I was interested in, and I looked at the data, I would say, this is pretty impressive, but it's a screening. And I would say, they've convinced me to the level that I will invest time in my lab to try and validate it for my use, or I don't get this. There's no proof here. But most of it's actually quite nice, and it would encourage you to get the antibody and work it out in your own lab, but it's not the final validation. There are other NIH-supported antibody portals that provide varying levels of characterization or validation. They tend to be more specific to an institute like cancer, or some are general. As we've talked about the developmental studies, hybridoma bank. There's one in DDK that's very specific for diabetes. There's really, we lack a cohesive general space to go to in this whole field. So I just want to finish, and there may be some time for questions, with some overall perspectives from NIH and then where we could use your help. And the first is that we don't have regulatory or policing authority. But what we can do with standards that the community agrees to, we can bring those to journals, we can bring those to our review process. So we need those. And so we do believe that we can intervene at the peer review criteria step. We can also encourage journals to set up their own steps for what standards they'll require for publication and for their reviewers. I think it's important that we've taken the position that even if you buy something that's validated, you have to validate it in your own lab for your own use. And again, as FDA would say, validation occurs in the context of its intended experimental use. And so we're looking to the scientific community to develop these consensus standards that we can apply, particularly in the review process, and that's the applicant knows what they need to do, it's the reviewer needs to know what they're asked to look at, and it's our program staff to work out a plan with the applicants if a grant were to be funded. So I'm going to leave it at that, Len, and if you want to entertain questions, okay.
There's time for questions.
I'm Carla Daniels from the DSHB, and I'd just like to extend one omission on your last slide. The DSHB is the clearinghouse for all of the hybridomas developed by the PCRP project and also the Cancer Institute. So you can go to our website, you can buy the hybridoma from us, or you can spend $40 and test it in your own lab. And it's been a very good, although unique, business model that is, I think, served the scientific community very well. I work very closely with Tara Hiltke at the NCI and also with Ignacio Pino at CDI for the PCRP project. And I think it's been a really great use of federal funds and NIH funds and has been very fruitful. It starts out slow because there are no citations. These are antibodies that are not tested. But slowly, over the last 3 or 4 years, we're gaining more and more citations, and I think eventually this is going to come to be a very fruitful project.
I want to comment that it is a very valuable resource. When I look at this, I'm responsible for the division that looks at trans NIH research programs like the Common Fund, Women's Health, AIDS, on and on and on. And the problem that we often face is that our approach is very distributed, and there's no single solution to the problem. It's solved all over the place. It's like the data problems we're dealing with is how do you make a coherent solution? So the most fundamental solutions that we can come up with here are going to be the most useful to NIH broadly.
Hi, thank you very much for your talk. I think this was great. The one thing that you kind of said a couple of times and then you reiterated, so I just want to kind of underscore this. The scored versus unscored criteria of this authentication document. So unlike the data management plan, which now people just click, download, and throw it in their grant and never think about again, this is actually looked at, and there's an acceptable, non-acceptable component to this, and you could confirm that. And so it's great. It's something that people are actually looking at because this is something that NIH can really, really affect everybody in this room about in terms of, is this something that people are going to pay attention to, or is this something that they're going to ignore as a new criteria? So I want to applaud you for actually putting that in as a, shall we say, semi-scored, but something that's paid attention to on the review criteria. So thank you for that.
We have quite a number of things that will prevent you from receiving a grant if they're not addressed, but they're not scored criteria. Human subjects, protection issues, and so on. So this is just added to that list that has to be resolved before a grant's funded. I guess one concern I have is, a reviewer could say it's adequate, but they might not know what they're talking about.
On the site.
Yeah.
In the beginning of the talk, you mentioned the increased visibility of the problem of reproducibility in popular press, in the media, right, in the John Oliver show. Do we have any sense, so it's indisputable that there's more visibility of it, but do we have any sense how the level of reproducibility has changed over the last 30 or 50 years? Do we have more or less reproducibility now? Because we haven't done the Begley, Bayer, and Amgen studies 30 years ago.
I don't know. Is there anyone else who can address that?
It's a very hard question.
Yeah, Len would know. He's saying that we don't have the data.
Right. And let's keep it in mind. So we may actually be at a place where we're publishing more reproducible research today than we were 50 years ago. It may be. It doesn't mean that we shouldn't do everything we can to make it more reproducible, but then let's not say that we're wasting 80% of taxpayer dollar because over the last 100 years, that same messy, irreproducible research has given us some amazing cures, vaccines, and progress. So let's just keep that in mind.
All right.
Jim, I just wanted to support some of the things you said. In a comment I posted here, I talked about Marshall McLuhan said, the medium is the message. And Cell changed the medium by putting the methods section in the back. And that changed the message, which was methods aren't important, right? Before that, you knew that, okay, the introduction talked about what, you were going to work on. And then the methods, you had to read the methods because before you understood the methods, you wouldn't know if the conclusions were right, okay? And so now the methods aren't even in the back anymore, they're in the supplement, and they're very hard to find. And I think that journals have pushed this in an unfortunate direction. The other change is that journals now make you publish a whole story. That was something Cell also promoted. And I think that often, people try to provide that last bit of data. And that's where they really strain. And that's, I think, where, caution can be thrown to the wind because they feel this enormous pressure to get that last little thing that completes the story. And I'm just publishing a paper now where we found the correct function for a protein, and there were 2 other knockout publications, and they each had an IP followed by a Western blot, and they each came up with an association that was contradictory to one another and contradictory to what we found.
It's the additive cell. Is that you can forget it.
Okay, let me, can I just add one more? Okay, I'll let go being blinded. All right, so when I was a postdoc, I went over to England to work on a factor that turned out to be a fictioner. And at that time in immunology, there were lots of people working on things that, have fallen by the wayside. Among them were antigen-specific suppressor factors. And I came back to Harvard. I was hired by Professor Benasraf, who was working on antigen-specific suppressor factors. And I knew that a counterpart of them didn't work, so I was very skeptical. And I said, okay, they wanted me to do the biochemistry on it. And I said, okay, I'll do the biochemistry on it, but I want to make sure that everything is blinded. So when I hand it, my results to you, and you do the functional assays, we'll unblind it at the end. And I actually didn't want to do the experiments because I knew it wasn't going to work. And in the end, they said, well, we're insulted that, you don't trust us and you want to blind it. So we're not going to, so you don't have to do the experiment. And they kept on publishing on it. And all the papers, are nonsense. And I don't know what it is, but I think one is not blinding. So experimenters read the results. A lot of it was just wanting the results to come out the way you wanted. So when you measured the thickness of the mouse's ear, you sort of adjusted, your calipers slightly, or, yeah, all right, I'm sorry. But blinding is important.
So, Josh LeVere from the Biodesign Institute. So one of the topics that's come up a few times here is, we've already talked about the fact that there are quite a few antibodies on the market that don't seem to be functional or are not reliable or have otherwise problems with it. And we've been talking about developing standards, and then the sort of subsequent question that comes to mind is, well, who's going to enforce that? So one concept has been, should there be a third party that, specialists who are, experts at executing antibody type experiments who could then validate experiments, publish those results, and in theory, if they did so, we could mark things on the market as either being valid or not valid, and therefore save people the trouble of ordering bad stuff, which in turn could save NIH money or at least allow it to apply its money in more profitable directions. So is that something that you could foresee the government ever supporting, or is that something that would have to have a, I mean, what, are there models that you could foresee where something like that could work?
I think you're talking about guidelines, and that's not something NIH will do. We are very reluctant to give clinical guidelines even. It's not in our purview. But I think the appropriate place is different societies. You know, I've been into the GI world, the gastroenterology and the American Gastroenterological Association review the literature, and they publish guidelines. This is known to be effective. This is what we think you should do. There's no evidence for this. So I think this goes back to societies in their own bailiwick to give the guidelines for the people in their groups.
Well, I think we're saying, I think the idea would be that a group like this might publish the guidelines, but now we're looking for an actual laboratory to do the testing.
You know, yes. So NINDS has done that. They have paid for, they've gone through this literature that they have of promising preclinical leads and said, which do we really care about? Which would we like to see go forward? And in some cases, they paid for attempts to duplicate that. But I can't see NIH duplicating, we'd have to have $64 billion. Yeah, so I don't know that that's the way to do that.
Just make it very quick and then very last question.
Okay, I really appreciate what NIH is doing to support this. And my question is, are you working with the Wellcome Trust and other European funding bodies to align on what you are doing? Or how can we move this forward?
We talk to them, again, because we don't have any specific regulations. They don't either. We're not going to align our regulatory policies. We have the same meetings with journal editors. It's a parallel process.
I'll second that. I think the NIH has been incredibly responsive to this issue and has really shown leadership. And my question is a bit unfair, but it's one I sympathize with. I'm Veronique Kiermer with PLOS, so I represent journals, and I think we're having a similar problem in the sense that with these new guidelines about rating grants, etc., what are the means that you have at your disposal to try to make sure that the study sections are actually following through in a way? And you sort of raised that issue saying, you know, perhaps this person doesn't have the expertise. I'm even sort of taking it one way further. What stops them to just sort of look at the number of Nature and Science papers that have been published in a previous grant to decide of renewal or something like that? What are the means that you have to really sort of have this kind of leadership penetrate to the level of the study section and the people who are actually rating these grants?
Okay, so I can give you some inside data from our portfolio analysis group. Is when we look at grant reviews, we do see statements of they publish in high impact factors, journals. We see that influences the priority scores. So it's our perception that people are using journals and impact factors as a surrogate for quality and using it for making their funding recommendations. That is why we've spent 3 years validating a new metric. I encourage you all to look at it. It's been pretty popular, actually. The relative citation ratio, which is a metric to get at the influence of an individual article. Regardless of the field, regardless of the journal it's published in. And we're hoping, actually internally, we're starting to use metrics like that instead of these other. But I wouldn't let the journals off the hook here.
So, Len, Jim, you've presumably seen Len's article estimating that $28 billion a year is wasted on research. That is irreproducible. I'm just wondering if what you think of that figure and whether NIH has done any internal studies to estimate how much of the money that you give out is essentially wasted, to use that word.
We haven't done an equivalent, no. I couldn't give you a number of an estimate of what we think was wasted money.
I mean, you see it as a serious problem, though, I presume, or not?
You just have to look at the results published by others and conclusions reached by other legitimate investigators and Len. Look, why are you here if you don't think this is an issue? And I want to end with one other thing. Folks, we owe it to our patients to take this on and do something, provide good recommendations from this workshop. So thanks for being here.
Thank you.